So our group members are Adila, Rawani, Amanina, Farhana, and Aisha. So what is predation? Predation is the transfer of energy and nutrients in community. The example of predation is herbivory, carnivory, and parasitism. Predator numbers are depend on prey, and predator may regulate number of prey.
So, the mechanism of plant herbivore system. So, the defoliation and consumption of fruits or seed. So, what is defoliation? Defoliation is a disruptive of plant tissue like leaves, stem, and root. So, feeding on seed can be detrimental, but it still help their dispersal, like their seed, mostly via feces. After that, the plant response after defoliation. It consists of 4 responses. So the first one is plant reduce available resources to reproduction. For example, if a plant has eaten by herbivore, it uses more energy to repair its own leaf. So the second one is in coniferous tree, like pine, have a high potential of defoliation and can cause a death. Okay, for example, when defoliation happens due to insect disease or pollution, the tree is lose its ability to produce food like sugar. So, without enough energy, the tree will die. So, the third response is increased attack by insect and disease, and the last response is the defoliation can cause changes of hormonal growth and regulators. The hormone like auxin, cytokinin, and gibberellin. The plant response consists of two types of response, which is positive response and negative response. So the first example of negative response is plant response after attack by predator like moth. The leaf becomes smaller and the canopy area reduced as much as 30% to 60%. Okay, so some trees end up with only 20% to 40% of original leaf. So the second example is aphid. Aphid can suck a sap from the plant and can cause reduction in growth rate and biomass, such as leaf, branch, and stem. And the last one is the positive response. The positive response. Okay, so the defoliation can be beneficial for some grass, where it greatly stimulates production by removing all the tissues that have low rate of photosynthesis, so it can expose a younger leaf to light.
Now, I will explain the different types of chemical defenses used by prey to avoid being eaten by predators. The first example involves venoms and pheromones. Snakes are a common example. They use venoms not only to catch their prey, but also as a defense mechanism to avoid predators. Another one is the sea lamprey, a type of fish that release pheromones into the water. These chemicals act as alarm signals and trigger fright and flight responses in other members of species, helping them escape dangers. The second example is secretion-based defense. These include animals like arthropods, amphibians, and reptiles, which produce secretions to repel predators. These secretions are often very strong smelling and can be detected easily. Some, like millipedes, release slimy or mucus substance directly from their body wall. Others, like beetles, may release chemical into the air, while some, like grasshopper and sting bugs, actually spray their chemicals toward the predators as a defense. The third example is based on odor and toxic substances. Animals like skunks and shrews produce secretion that smell terrible and discourage predators from approaching them. Also, many arthropods and even some caterpillars, like the monarch caterpillar, can store toxic secondary compound from the plants they eat. For example, monarch caterpillar feed on milkweed, which contains cardiac glycosides. When birds eat them, the toxins make birds sick or even cause death. These toxic substances include alkaloids, terpenes, glycosides, and tannins and are found in both arthropods and plants.
Okay, next we go to mimicry as a prey defense strategy. Okay, what is mimicry? Okay, mimicry is when one species will mimic resemble another species, the model to gain an advantage, usual to avoid predator. This strategy works best when both the mimic and model share a similar habitat, and predator have learned to avoid the model. So, essentially, it is a survival through deceptions. Mimicry is only visual. It can involve smell, behavior, or movement. It is play a role in co-evolution where multiple species influence each other developments over time. Okay, next we go to predation mimicry. Predation mimicry is like bluffing a harmless species imitates the harmful or toxic species, so predator leave it alone. For example, the ash borer move look like a bee. Big stealing so predator avoid anything that look like them, even the harmless move. This only work if harmful models are more common than mimics. If too many mimics as predator might stop believing the warning. Okay, next we go to Mullerian mimicry. In Mullerian mimicry, multiple harmful species involved to look alike. They team up by sharing warning signal, helping predator learn faster to avoid them. A good example is Viscerori butterfly and Monash butterfly. Both are toxic to birds, so their shared appearance strengthens the predator memory. Okay. This is a mutual reinforcement. Everyone benefits by reducing theories. It's a great example of convergence evolution, similar traits arising independently in different species. Okay, next we go to plant insect mimicry. Plants are not left behind in the mimicry game. Some, like the passiflora plant, mimic butterfly egg on their leaf. Female heliconius butterfly avoid laying egg, where others already have, so the plants get protection by pretending it's already taken. This is called egg mimicry, and it is a form of visual deception used to reduce herbivore. Okay, next we go to cross-lineage mimicry. Okay, sometimes mimicry happen across totally unrelated species. This is called cross-phylogenetic mimicry. For example, some caterpillars have an eye spot that look like snake eyes to scare off predator. Even young lizards or snakes mimic dangerous millipedes. Okay. This mimic often use startles display surprising the predator just long enough to escape. The mimic and model may not even be close related, but similarity is enough to confuse predator.
Prey defense, armor, and weapon. Animals like clams, armadillos, turtles, and beetles withdraw in their armor, coat, and shells. While porcupine, echidnas, and haddock have modified hairs called quills. It is sharp and painful. Prey defense, alarm call. Alarm call attract predator attention away from conspecific or could attract more conspecific for cooperative defense. Example, Belding's ground squirrel. This function to warn close relatives. And harassment of owl by small birds, bring in number of potential prey that respond by mobbing, grouping, harassing predator. Prey defense, predator satiation. Synchronization of birds or eggs to reduce predation of newborn. Most of offspring are produced in a short period of time to enhance their numbers. One strategy employed by caribou and wildebeest. Production of increased number of prey so that the predator only take a fraction of them.
Today is a warning coloration. Warning coloration offers approximatic coloration. Animal that possess pronoun toxicity and another chemical defense often possess a warning coloration. Usually they will have a pole color that very bright and noticeable or design. Cryptic coloration is also called concealing or protective coloration. It helps prey hide from predators. The prey blends in by using patterns, shapes, posture, movement, and behavior. This defense is common in fish, reptile, and ground nesting bird. Stick insect use this by looking like stick or twigs.
Some prey animals, such as a butterfly, grasshopper, and ungulates, use flashing coloration as a defense mechanism. The animal displays sudden visible color patches when they are disturbed, which can startle or confuse predators. The color may distract or disorientate predators. In species like deer, flashing color may also serve a social function by helping maintain group coordination when threatened by a predator.
Our objective for this lab is to calculate and describe the following descriptive statistics, which is mean, variance, and standard deviation. The second one is to understand the concept of correlation, and the third one is to determine the relationship between the two variables.
The result of set 1. The leaf length has a mean 25.25, variance 4.13, standard deviation 2.03. The leaf width has a mean 5.76, variance 0.57, and the standard deviation 0.25. The leaf weight mean 1.58, the variance 0.09, and the standard deviation 0.28.
Result set 2. The table shows the leaf length, leaf width, and leaf weight. The leaf length, the mean is 19.93, the variance is 7.0, the standard deviation is 2.65. The leaf width, the mean is 8.31, the variance is 1.33, and the standard deviation is 1.15. Lastly, the leaf weight. The mean is 1.79, the variance is 0.13, and the standard deviation is 0.36.
Next, we go to result set 3. Okay, the leaf length for mean is 60.50 cm. The variance is 5.9328. The standard deviation is 2.435. Okay, next, leaf width cm. The mean is 5.52 cm. The variance is 0.4684. The standard deviation get 0.6845. Okay, and then leaf weight gram for the mean get 1.473 gram. The variance is 0.1909. The standard deviation get 0.437.
The result of set 4, which is left length. Left length has a mean of 22.95 with a variance of 27.05 and a standard deviation 5.20. Next is left width. Left width show a mean of 9.30 with a lower variance of 2.26 and standard deviation of 1.50, suggesting that the width are more consistent compared to the length. Last is leaf weight. Leaf weight has a lowest mean of 2.38 with a variance of 0.96 and standard deviation of 0.98, indicating the least variant among the three traits.
The graph shows the length of the leaf and the width of the leaf. There are 4 sets, set 1, set 2, set 3, and set 4. The graph shows a moderate positive linear relationship between the width and the length of the leaf. As the width increases, the length also generally increases. let's move on to the lab 2. Factors that affect the distribution and abundance of organisms. Objective is to introduce the simple concept of quadrat, to enhance the power of observation, to record the difference in plant distribution within the quadrat, and lastly, to infer from natural observation the possible factors that affect distribution and abundance of organisms. The method. In practical class, one type of forest at UiTM Jengkel has been area under the study. The second one quadrat of 5 meter times 5 meter was made at the forest. Multiple quadrat sample was performed throughout the habitat at several random locations. The third one, all tree stands that are found inside quadrat were observed and were taken note. Lastly, data were used to estimate the population size and the population density within the entire habitat.
Next is the result of quadrant 1. In this quadrant, we can observe the distribution of 8 different species. Some species are widely spread, like species B, and some are in small groups, like species C and species E, and others are very rare, with only one individual, which is species E, G, and H.
Okay, next we go to observation for result 1. Okay, this result observation for the result quadrat. Okay, the quadrat have 8 species. Okay, first species species A. The symbol is asterisk flower. The count is 1. The percentage is 5.6%. Okay, and then species B, the symbol is star, count is 1, the percentage are same like species A, 5.6%. Okay, next species C, the symbol is cloud, count is 4, the percentage is 22.2%. Next, species D, the symbol is triangle, the count is 3, the percentage is 60.7%. And then the species E, the symbol is flora emblem. The count is 6. The percentage is 33.3%. These species are very high than other species. Okay. Species F, the symbol is gyre. Count is 1. The percentage is 5.6%. They are same like species A and B. Species G, the symbol is Christian's moon. Count is 1. Same like species F. Species H, the symbol is circle dot. Count is 1. Same like species G.
Observation quadrat through, we get 5 species. First one, species A. The total number of individual we get 6. Species B we get 1. Species C we get 4. Species D we get 1. And last species E we get 10. So the total number of the individual we get 22.